start now. So I'm very glad to introduce the first speaker of this year, this new academic year, with Maira Aguiar from the BECAM, which is a Basque Center for Applied Mathematics in Bilbao, in Spain. And she will talk on the origin of complex dynamic in multi-strain dengue model. So please.
Yes, thank you very much, Pierre, for invitation. As I said, I'm glad to, I need to be able to share my screen.
Yes, sorry.
Host is disabled.
Forgot. Let me see. Yes, here it is. Here it is. Sorry.
Oh, yeah.
I forgot that.
All right, thank you.
Very nice. We can see your.
Okay, I guess you can see my mouse and my screen, right?
Yes.
Okay, so again, thanks a lot, Pierre, for this invitation. Sorry that I could not join 1 hour earlier, but I had, a commitment that I could not postpone. Thank you for attending. I know that everybody's very curious and tempted to hear about COVID modeling. I was requested to give a seminar, and indeed, I'm working on COVID, but I have restarted my preliminary work on dengue, and it's very interesting. So I thought that we could change a little bit the subject. So I'm going to talk about multi-strain dengue models and focused on the dynamical systems, or dynamical systems theory, to look at the complex dynamics one can find in the simplest models. And of course, the finding of this chaotic dynamics in this kind of models have a major role on the epidemiological scenarios and interpretation of public health interventions. We are going to discuss a little bit. I will try to keep the time. So just a brief overview. I guess most of you, especially in Asia, heard about dengue already, but it's important to mention that is a viral mosquito-borne infection, which is the leading cause of illness deaths in tropics and subtropics, although it's no longer restricted to the tropics and subtropics. This map here shows the risks of acquiring dengue via the force of infection, published latest in 2013. The situation didn't change much, but as we were discussing earlier today, there are already dengue vectors identified and established in some European countries, and local transmissions are already reported. So it's just a matter of time where it's becoming also a problem in European countries. For now, we have more than 1/3 of the world population living at risk of acquiring dengue infection, which makes this disease a major public health concern. Dengue is very special because although it can be very mild in the first infection, you can acquire more than one infection, and the secondary infection have a higher probability or higher risk of developing severe form of the disease. So it is special because there are 4 antigenically distinct viruses, but they are very closely, and that gives a problem within the host, within the immunological response that I'm going to describe in the next slide. This figure here just to show the difference on virus identification or presence between 1970s and up to date. And more important than look at the 4 serotypes have been spread and established in different areas of the world very quickly, to notice that in the Southeast Asia, the 4 serotypes are coexisting at least since 1970s, which makes this region a very interesting region to study, where we can find the coexistence and the data on this coexistence. So to model dengue fever, we need to get a little bit of knowledge on the epidemiological or on the biology of the virus and the transmission and the infection. We know that infection by one serotype, you confer lifelong immunity to that serotype, which means that once you are infected with one serotype, you can no longer be infected again with the same serotype. And you also have what they call temporary cross-immunity to the related serotype. So you have a period of time that you are protected against all related serotypes till these levels drop and you become susceptible again with a historical pre-existing infection, meaning that you have a pre-existing specific antibody for a given serotype. Dengue has a wide spectrum of clinical presentations. Most of the infections are asymptomatic, but it can range to mild and severe cases. And as I said, epidemiological studies have supported the association between severe disease and secondary dengue infection due to a process called antibody-dependent enhancement. that I try to explain here. I'm not going to take too long, so we can discuss a little bit more during the questions. The difference between primary infection and secondary infection within host in terms of probability of acquiring severe disease. So most of all the infectious diseases start, the immunological response start with an unspecific antibody called IgM, which is generated upon antigen presentation and is able to bind it to this virus and clear the infection. While the body is fighting against this infection, you have also generated a specific antibody or memory antibody called IgG. And this IgG is then conferring the lifelong immunity against this given serotype. The level drops while you have this temporary cross-immunity period, and then you become susceptible again. And what happened in the secondary infection with a different serotype is that the body recognizes a new serotype as being the first one, so the previous serotype, and you have a tentative of clearing this infection using the memory and specific antibody rather than starting again from the beginning. And this generated a problem because although this antibody here, memory, can bind into the new virus, it cannot neutralize the virus. So instead of neutralizing infection, it enhances the replication of the virus inside the cell, and you have a higher viral load followed by an enhanced immune response, which leads to hemorrhagic symptoms. Very briefly, we can discuss that in detail if you are curious. The important thing here is that primary dengue infection, you have a normal immunological response mediated by IgM, then IgG, and secondary infection with a different strain. We start with IgG. The IgG is not able to clear the infection, so we have to start again from the beginning with IgM and then IgG. And this shift will allow the virus to replicate more rapidly inside the human body, which leads to severe symptoms such as hemorrhagic symptoms. Okay. We know that the majority of secondary dengue infections occur at a space of more than 6 months, which means that you do not have the same individual acquiring dengue 2 times in an interval before 6 months. So if you get now, and you are living in an endemic area with a different serotype circulating, you can get a secondary infection, but that could take at least a year or so, not only because of the temporary cross immunity, but also because of seasonality. Dengue is a very seasonal disease. We need vectors to transmit. Yes? Is that okay? Okay.
Good.
Okay, so there is no specific treatment for dengue, and all the severe cases which show some severe symptoms would require hospitalization, which means that basically all the incidence data we have are people experiencing at least 2 infections because they have shown some symptoms. As I said, primary infection are often asymptomatic. So secondary infection can be mild and severe. People are admitted to a hospital, and that's the data we have to work with. So we have to take into consideration this fact. Apart from there is no treatment, specific treatment, we have vaccines. The first licensed was produced by Sanofi Pasteur, it's called Dengvaxia, was licensed in more than 20 countries after the phase 3 trial was completed. But since 2016, we have had some fights in how best to use the vaccine, because we could see that vaccine efficacy, first of all, is not static, and second, has a really a problem regarding the immunological background of the individual receiving this vaccine. For instance, we show here the vaccine efficacy for this Dengvaxia for individuals younger than 9, and we see that for the first 2 years, you have a stable vaccine efficacy, which drops in the year 3 to a negative value. Most of the confidence interval is in the negative value, which means that vaccine generates more dengue cases than non-vaccinated people. However, when you look at individuals which are older than 9 years old, we have, again, these 2 years of stable vaccine efficacy, dropping in the year 3 with a bit of confidence interval to the negative, but kept, that means within the positive vaccine efficacy, which means that for this age class, you would have a small beneficial use of this vaccine. What we didn't know, and we are going to show it in the next few slides, is that the youngers were observed to be seronegative, which means that they didn't have a previous infection before, while the older were seropositive, which means that they have experienced already a primary infection, which means that the vaccine was basically boosting the immunological response rather than in a younger seronegative that was kind of mimicking the primary infection and enhancing the risk of being severely ill after they acquire a natural primary infection. Hope that is clear. Okay, and then we have a second vaccine that was much expected, the produced by Takeda vaccine. We could evaluate the trial, and in this case, the vaccine producers had a bit more chance to explore the ways of performing this trial. So they have tested everybody beforehand who were seropositive, who were seronegative, and their vaccine apparently shows the same problem that the vaccine, for instance. Between seropositive and seronegative, you see that seropositive has a much higher vaccine efficacy, close to 90%, while individuals seropositive would have a negative vaccine efficacy. So we should be careful with that. Also, we observed that as time passes, the overall vaccine efficacy has decreased. So this is the time between 12 months and 18 months, going to a lower vaccine efficacy overall. We could evaluate also the vaccine efficacy for per serotype, and here I compare the Sanofi vaccine and the Takeda vaccine. And what we see here, the Sanofi vaccine has two groups of vaccine efficacy per serotype, where dengue 1 and dengue 2 had a small vaccine efficacy compared to dengue 4 and dengue 3, which was overall, which was a bit higher. And the similar behavior we found for the Takeda vaccine with dengue 2 as a very good vaccine efficacy, but that can be explained because both of the vaccines are chimeric, so they use backbone of other virus in order to prepare to produce the vaccine. In the case of Takeda, the backbone was dengue 2, so was expected to have up to 100% vaccine efficacy against this serotype, but for the other serotype, vaccine efficacy varies. And when you look at it, we just say, although we need a tetravalent vaccine to avoid antibody-dependent enhancement and severe case of dengue, none of these vaccines are a tetravalent vaccine in the sense that you have different vaccine efficacy by different serotypes. So that also has to be taken into consideration. Okay, but we are going to discuss how to model dengue best. And of course, the model framework will be defined by the type of data and the question you have. So this is just to illustrate the type of data one would have for dengue. This is serotype data for Puerto Rico. We have mosquito eggs in Brazil. We have incidence data, in this case here for Thailand. This incidence data can be also stratified by geographic regions. This is a map of Thailand with the 77 provinces, and we can see where we have higher incidence going on. And we have also the so-called serological data, which counts the number of viral particles one have during the infection and how it wanes while the immune response is acting. But we are talking about complexity and how much complexity do we need to describe dengue dynamics or any kind of question regarding to within-host modeling. This is just to illustrate recently published, sorry, I should have corrected here. This is just published in Biology, where we have a very simple framework to describe primary infection, secondary infection with the same virus, and secondary infection with the different virus. And we see that the model is very good in terms of describing the immunological response. For instance, let's concentrate in this red curve here where we have the viral particles for primary infection to a certain level. In a secondary infection with the same virus, we expect that because of the pre-existing antibody, you would have less viral load, and that would eventually be the threshold where people do not have any symptoms. I recall that symptoms are correlated to amount of virus you have in your blood sample. And then for the secondary infection with an heterologous virus or with different virus, you have a higher viral load than in a primary infection, and that would eventually be correlated to severe symptoms. So this minimalistic framework, although does not show any complex behavior because it's just one curve, can describe already the antibody-dependent enhancement, can evaluate the role of temporary cross-immunity and the impact of vaccination once it is validated with data. So we are working on that. Now, what about the disease transmission? Can a simple SIR be helpful in order to understand the transmission of dengue? How much complexity one would need in order to describe this type of oscillation shown here in red, which is my data. In blue is the model simulation, and in red is my data. how much complexity do I need to put in my deterministic model in order to observe this kind of oscillations? And for that, we would need to take into consideration 5 main ingredients, which are listed here as model parameters. We have infection rate, we have recovery rate, and we have demographic rate, which are all well-known parameters in dengue epidemiology. And we end up with 2 parameters here that there is no animal model to describe, but we have to estimate from the data, knowing the range where this parameter should be. One of them is the temporary cross immunity, so how long after primary infection individuals are protected and able to become susceptible and get a secondary infection. So that's my alpha. And the phi is like, what is the contribution of individuals experiencing a secondary infection if they have higher or lower viral load, if they are hospitalized or not. So this phi here would enhance the transmission in a secondary infection or decrease the transmission in the secondary infection as compared to the baseline transmission for a primary infection. Okay? We can write down a system of ODE. Looks terrible, but it's very simple. I just divide the population into 6 large groups. I have seronegative individuals, so those individuals which have never been infected before. Individuals acquiring a primary infection with strain I, which means dengue 1, 2, 3, or 4. So I could be from 1 to 4. This individual recover from the primary infection, and after a period of temporary cross immunity alpha, becomes susceptible again, but with a pre-existing antibodies for the strain of a primary infection. This guy here can acquire a secondary infection with strain J, and to be realistic, J must be different from I because of the lifelong immunity against that serotype. And if this guy survives, he recovers. I only consider 2 infections because we do not have data on tertiary quaternary infections, and even though from cohort studies we know that the tertiary quaternary infections are super low frequencies because of the antibody-dependent enhancement that boost your immune response and you are basically protected forever. Okay, so I started analyzing the complexity of the model from the most simple one to the most complex one. And the simplistic version would be to assume primary infection and secondary infection. This is not realistic for dengue because I have here no strain structure, but I have susceptible individuals or seronegative individuals becoming primary infection and then recovery, and then after a period of temporary immunity, acquire a secondary infection, become secondary infected, and recover. Important thing here is the parameter phi that I put next to the secondary infected, and I assume that individuals in the secondary infection would transmit the disease a bit less than individuals in the primary infection, so my phi would be a smaller one, because this guy would have some symptoms and would be in hospitals, which would decrease the probability of this guy transmitting the disease in compared to this individual in a primary infection that is mostly asymptomatic and still run around and transmit the disease. Okay, but we can analyze this phi in different parameter values just to see which kind of dynamics one get. Again, I have no vector dynamics, so seasonality would mimic the abundance and the decrease of vectors and only 2 possible infections. And I compare here the results I have for the non-seasonal model and the seasonal model. I fixed my alpha equals 52, which means that individuals are recovered from a primary infection and immediately able to get infected again, which means I have no temporary cross immunity. And my phi is fixed to much larger than 2. And I say that's why, because phi below 52 and alpha below 52 and phi below 2.6, I have nothing but fixed point in the non-seasonal model, and I have a limit cycle in the seasonal model, okay? So complex dynamics or any interesting dynamics only comes up when I fix this. Again, not realistic for dengue because here I neglect temporal cross immunity and I assume that individuals in the secondary infection transmit the disease much more than individuals in the primary infection. When I fix the parameter here, I have a hope verification happening for temporary cross immunity around, so 52 is 7 days, 50, that's 15 days or so. And as soon as I add seasonality, we get the complex behavior here. I'm looking for the temporary cross immunity. When I look at the phi, the ADE ratio, I get again hope verification happening for phi much larger than 1.5, and here only fixed point, you can see here. And as soon as I add seasonality, I get the complex dynamics, the chaotic behavior for phi much larger than 1.5. Okay, I can look at the variation of seasonality, variation of imported cases, and we look at the so-called Lyapunov exponent, that's the eigenvalue we basically have for the systems with more than 3 dimensions. And this is the Lyapunov spectra where I vary temporal cross immunity and the AD ratio, showing that I only can get complex behavior in these models when I assume that there is no temporal cross immunity, so phi alpha is much larger, and phi is much higher, which means that secondary infection must transmit much more than individuals in a primary infection. So this is the output the model gives, and for in this case, we don't even need any kind of statistic methods to reject this model. The data is in red, the model is in blue. We have irregular outbreaks every 25 years, and it cannot describe dengue dynamics. Okay. Okay, so we can add more complexity. In this case here, I add 2 equations for the vector and SI. And remember, and I have seasonality again. Remember that we had, let me see if my computer allow me to go back quickly. I have so much to tell, but the time is short. So you see here that we have a bifurcation, field bifurcation diagram with complex dynamics, which is confirmed by the positive Lyapunov exponent for this model. As soon as I add 2 more equations, you have a gap here, and we don't know what's going on. So the vector dynamics in this case is stabilizing some of the chaotic dynamics, and we are still investigating that. But when you look at the output, we see that still this model can be rejected because I cannot describe the data either. However, the Lyapunov spectra show that it moved, the complexity dynamics moved from here to here, which means that now temporal cross immunity is considered important. I don't know what the vector is doing, and we are investigating that. I'm super curious. I hope not to lose the chaotic behavior. Okay, second step is to add strain structure. And I have the similar dynamics as I presented before, but now I have 2 different strains. So individuals can get primary infected with strain 1 or strain 2, recovered from the primary infection with strain 1 or 2, and after a period of temporal cross immunity become susceptible again with a pre-existing antibody related to the first infection, and get a secondary infection with a different strain than the primary infection. So if this guy gets a primary infection with strain 1, can only get infected with strain 2 and vice versa. Again, 2 infections, no vector. This is the system, looks horrible, but it's super simple as well. So I have seronegative, primary infection with strain 1 and 2, recovered from strain 1 and 2, temporary cross immunity becomes susceptible again with a pre-existing antibody, and then you can get a secondary infection. My phi here as well was assumed to be smaller than 1, which means that I assume because of hospitalizations, individuals in the secondary infection would transmit less the disease than asymptomatic individuals in the primary infection. And we can just confirm that phi below 1 is really important. We have here the bifurcation diagram varying this ADE ratio for no temporal cross immunity and 6 months of temporal cross immunity. And we see that when we neglect the temporal cross immunity, we only get a chaotic behavior for phi much larger than 2. However, as soon as we add this extra feature into the model, a new chaotic window is observed, where no longer we need to restrict the fact that individuals in the secondary infection, hospitalized individuals, would transmit more than individuals in the primary infection. So we found out for the first time, that was in 2007, a new chaotic window for phi below 1. And we kept analyzing that. We confirmed that it's chaotic by the Lyapunov exponent. So here we have a Lyapunov spectrum with a positive Lyapunov exponent. And please notice that is already non-seasonal model. So I have not much external forces going on, but still a rich dynamical structure from Hope bifurcation, pitchfork, torus, and tangent bifurcation. Was the first time that torus bifurcation was found to be the root of chaos in an epidemiological system, which brings a lot of interesting aspects to discuss in terms of intervention measures. Without seasonality, that's the output we have. We have a regular pattern every 5 years. And as soon as we add seasonality, we get a realistic pattern with irregular and yearly outbreaks, which can be matched with the data. Of course, remember that's chaotic, so I cannot have exact the 30 years of matching data, but I have patterns repeating, and we have then to evaluate what is the limitation in terms of prediction this model gives. So the simplest 2 infection with strain structure model gives already a qualitative very good result when comparing empirical data and model simulation. We played with the Lyapunov exponent. Here, I showed the seasonal already because we need the seasonality to match the data. I fixed my phi equals 0.9. What I assume here is that individuals in a secondary infection would transmit 10% less than individuals in a primary infection. So where it is here, we have a dominant Lyapunov exponent of 0.12, which gives approximately 8 years of prediction horizon. And this is only to show the exercise where we have the attractor of our model. And then I perturbed a little bit the system with a small change in the initial conditions. And I run again. So I plot the second trajectory on top of the primary from the first trajectory. And I have that indeed 1, 2, 3, 4, 5, 6, 7, 8, 9 years. I can match both of the trajectories. So I have a good prediction. However, after this time, the system is completely unpredicted. And that's the characteristic of deterministic chaos. You have a short-term prediction horizon and a long-term unprediction. Okay. Okay, so we did a lot of research. We had to compare what would be similar between 2 strains, 3 strains, or 4 strains. And what we could observe is that independent of the number of strains, we always have restricted to have complex dynamics for assuming alpha to exist. So assuming that there is a temporal cross-immunity. When you neglect the temporal cross-immunity, you get nothing but fixed point and limit cycles. And the attractors are more or less the same when we have the same parameter set. So that was found that the strain structure of pathogen shifts the chaotic behavior to a more realistic parameter region and is not related to the detailed number of strains, but rather the combination of temporal cross-immunity and antibody-dependent enhancement parameter phi. So we compare the 2 strain versus the 4 strain dengue model. The dynamics exactly the same. The difference here that you have more combinations of secondary infection. We have a dimensional problem. We jump from a 9-dimensional system to a 25-dimensional system, which is difficult to treat. But when you look at the output, the time series and the data matching are very similar. And quantitatively, the Lyapunov exponents and the bifurcation points occur at the very same place. So the dimension and the structure and the magnitude of the dominant Lyapunov exponents, yes, I got it, were the same. So we would not gain anything by adding more strains to the system, more complexity. But in terms of prediction, in terms of dynamics, it would be the same. So we just kept using this 2 minimalistic strain in order to include the vaccination, as I said. So we have developed an age structure model with the baseline of our 2 strain model, assuming the recommendation of the WHO and the producer. We have then groups of individuals younger than 9 years of age and older than 45 years of age that cannot receive the vaccine. Vaccine was not recommended. And then we have a group of individuals between 9 and 45 that can be vaccinated. So how do we implement this vaccine? Either individuals have never been vaccinated before, are seronegative and receive the vaccine with a vaccine efficacy new, or you have individuals which have been already infected before and can be vaccinated and move then to the recovery class because the vaccine, as we remember the graphic, boosts the immunological response of those who have been already vaccinated. What we have here, individuals below 9 and older 45 cannot be vaccinated, but can go through the disease stages because they are living in endemic country. They can get primary infection, secondary infection. However, those one here which are entitled to get the vaccine can either receive vaccine as seropositive, being protected with a vaccine efficacy and removed. So you decrease the hospitalizations of secondary infections here. However, if you vaccinate individuals which are seronegative, you mimic the primary infection. So you move them to another class called sensitized class. And as soon as they get the primary natural infection, they would have a probability of developing severe symptoms. So you have an increase of hospitalization here. Okay. So this is only to summarize what we have done on the Sanofi vaccine. We look at different vaccine efficacies, and what we assume is that individuals which are seronegative have a lower vaccine efficacy than individuals which are seropositive. We have our framework. We could match, we decided to match the 5 last years of data we had and introduce the vaccine and then see what happened in the next 10 years, knowing that our system has a prediction horizon of approximately 10 years. And what we see here is that when we do the vaccination without immunological screening, meaning that you vaccinate individuals between 9 and 45, so the green is the data, in red is my model without vaccine, and then in black is the model with vaccine. And after 5 years, you have an increase of 25% of severe cases in this population. However, when we look at the individuals, when we restrict this vaccine to individuals which are seropositive only, we see that we can reduce up to 40% the number of hospitalizations. So this graphic here shows both simulations. So in red is the model without vaccine. In black is the model with vaccine without screening the population. So we vaccinate either seropositive and seronegative. And in light blue, we have the vaccination only in seropositive individuals. Okay. I'm going to skip that. And I'm just going to give you an overview what happened on the dengue vaccine. So the vaccine was recommended by the WHO in 2016. And from the data from the phase 3 trial, we could have seen that already there was some problem with seronegative children. In the end of 2016 and 17, they implemented in 2 large vaccinations in the Philippines, Pierre, that you have worked, and in south of Brazil as well, which dengue is not a problem. And we kept publishing and telling them that they should be careful, they should screen people before that, till they start to do new tests on those who participated in the trials. And in fact, they have shown that seronegative had a higher probability to develop severe disease after being vaccinated. So the programs were suspended, but not before that more than 1 million children, where around 15% could be seronegative, were vaccinated. So in mid of 2018, WHO raised new recommendation, and this vaccine now requires a pre-testing. But it was a bomb that could have been avoided since 2 years.
Maira, I think it's good time.
It's my last slide. Okay, no problem.
I think your last slide was perfect to stop.
Okay, so just to show you here the data for dengue vaccine, that we see that indeed we have a negative vaccine efficacy observed here for seronegative individuals being vaccinated and then being infected with dengue 3. So this vaccine still not to be licensed. I start a new fight now because they are just finishing it and trying to license. Hopefully the people would be more careful. Thank you very much for your attention. Draw your attention to the MTB seminars. We also have one, of course, every other Friday, and our conference, which takes place next February. Please stay tuned and follow us on Facebook. Thank you. I'm happy to answer questions.
Okay, thank you very much for this very nice talk. No, please leave your slide, because probably we will have question about it.
So that you want
I think so. Anybody has a question? Please raise your hand if you have a question. We can, and I can give you the opportunity to ask some questions about this presentation. So, if nobody has a question yet, I will have some. So. First, at the beginning, you show several times some figure about vaccination, and with a difference between at the third year, right? You show several times from the beginning. I think you show some figure. Let me see. Maybe go down now. Yeah, this one. What's the precise meaning of this figure, in fact?
Okay, so this is the vaccine efficacy for CYD14, which means phase 2 trial that Sanofi performed in Asia.
Yeah.
And this is 4 years of data.
Right.
The Bayesian, we use the
Bayesian. So it means that you give one shot, then second shots?
No. The vaccine has 3 shots. So you give 3 shots to the individual, 0, 6, and 12 months apart. Then you follow the group in order to see how many cases you have in the group which received the vaccine and in the group which didn't receive the vaccine. Yeah.
Oh, I see.
The issue with the Sanofi is that they didn't pre-test individuals. So on the group which received vaccine,
Yeah.
and the group they didn't receive, there was seropositive and seronegative, but they didn't know who. Because younger individuals are seronegative more than older individuals. That's correlated because of time. So that was the issue. Takeda has a different issue. They tested everybody before, so they have a better knowledge of what's going on in their vaccine.
Right. I see. And so it means this shows a sort of log quantity, I assume.
No, this is the.
What's this number here? What the vertical axis? What's the meaning of the vertical axis?
The y-axis is the efficacy, which is the relative risk of the vaccine. 1 minus the relative risk of the vaccine is equal to the vaccine efficacy.
Okay.
So it's very
it's more anyway. So that's.
Let me show you something here. 44 maybe. Oh, why it doesn't go?
I have. No, that I think that's fine for the moment. Okay. Any other question among the? If you want, it's good time to ask question to Maira.
Here, while people think about it, this is the table for the vaccine trial on Sanofi. We have 3 sites, okay? We have CYD14 that is in Asia, the CYD15, which is in Americas, the CYD2257 is still the phase 2. We look at the data on CYD14 because it covers larger age groups. In the Americas, they didn't include children, yeah? And then here the data shows how many cases in the year 1 you have individuals receiving the vaccine and in the control, and then they calculate what they call in epidemiology
so it
can be transferred to the vaccine efficacy concept, where it's 1 minus relative risk. Okay. And that's the y-axis. It's here.
Okay, I see. So please don't be shy to ask question. So then I have another question. At some point you ask, you show some chaos or some complex dynamic, right? This was for the model with this kind of figure. Can you show the model again? I think it was OD model, right?
Yeah, non-seasonal one.
It's non-seasonal one.
Exactly.
And you get chaos. But you get the time-dependent term, right?
No, this one is without seasonality. When I get seasonality, this box is completely filled. It's chaotic everywhere. Without seasonality, we have 2 chaotic windows for different high ratios.
Right.
And some, coexistence happening here. When we look at this area, for instance, that was the first time described for 5 below 1, you have isolus happening here, you have Hope bifurcation, you have pitchfork bifurcation, you have torus bifurcation, you have tangent bifurcation, you have a super rich system. It is non.
Yeah. Can you show the system again, please?
Yes.
Because well, the point, you must have a reinfection somewhere, right?
I have a secondary infection. Yes.
Right, but no. Yes, the system here.
So please forget about this T here, because I just afterwards I need to add seasonality in order to match the data.
Yes. Yeah.
But the first bifurcation diagram is without considering the seasonality.
I see.
So I have primary infection, recovery, and the temporary cross immunity that it's a kind of delaying, but still basically with a simple parameter can be mimicked, and then you have a secondary infection.
Okay. So. Okay. Yeah. Sure. And this leads to I see. Okay. This leads to very complex dynamics. Very surprising.
Yeah.
Very
surprising.
surprising.
For us as well.
Yeah. And so you include a constant flux here, input flux of susceptible to, okay.
Everybody which dies
Okay.
and we have a constant population size, yeah.
Okay, so very interesting. So it's just a matter of having this multi-strain. Huh?
Exactly. Now, this assumption of ADE, this assumption of individuals in the secondary infection transmitting slightly less or transmitting differently from individuals in the primary infection is the key to generate this complex dynamics. Right. We have people publishing with this phi being enhancing the susceptibility rather than transmissibility, and they find this chaotic window here, but they do not find this. This one is missed and is related to the combination of phi and temporal cross immunity. Okay, I see. Oh, okay, that's pretty cool result. And okay, so I didn't know about this this kind of. Well, I can send you some references. We have that was basically my PhD, and it's really indeed really interesting because it's very simple model and gives already super rich dynamics. Oh, that's cool. That's cool. Yeah, I want to have some references. Oh, sure. I can about this. I include that. I will include that somewhere soon. Yeah, great. Happy to know that you got interested. It's really indeed very interesting. Yeah. Are there some people who wants to ask any question now at this stage? Oh, apparently people are a little shy to ask questions. You can email me if you want. Okay? Yes. So maybe we can stop here, and so I want to thank you again for this very nice presentation. And I make an announcement for the next week. Next week we will have a presentation from people from the group, a student of Hiroshi Nishiura, from a PhD student of Hiroshi Nishiura, will talk about some COVID-19 in Japan, probably. Huh? And so this will be the 2 talks of the month. Only 2 talks per month. I think it's good enough for this webinar. And so thank you very much for listening and coming, and thank you very much, Maira, for this very nice presentation. Yeah, thank you, Pierre, for the invitation. I'm just replying to a guest here, Dao Zhu. He asked, the 2-strain and 4-strain models give similar results. I wonder what is the difference in parameter setting between different strains. And I was telling you, there is no difference. We use the same parameter set. What we found out, and that is published in Ecological Complexity in 2013, that the complexity comes from the combination of ADE and temporal cross immunity rather than number of specific strains you had in the model. You can have as many strains as you want. If you do not have the complication of ADE and alpha, you have no complex dynamics. So you need to have this combination. Okay? The parameter set is the same. So thank you very much. Yeah, welcome. Okay, see you next week. Bye-bye. Bye-bye. Bye, everybody. Thanks for listening. I stopped to record. Oh, yeah, you have to.